So carbohydrates, this part of medical microbiology, we call it the biophysical chemistry, where we talk about biomolecules, water, carbohydrates, proteins, lipids, enzymes, and nucleic acids. So carbohydrates are one of the most widely distributed biomolecules in both plants and animals. Actually, the most abundant biomolecule. If you hear of a molecule known as biomolecule, it's a living molecule. Okay. Now, carbohydrates are made up of they have got what we call a carbonyl functional group, a carbon double bonded to an oxygen. If that carbonyl is at the end of the chain, we call that as an aldehyde. If it is on any position other than the end of the chain, we call it a ketone. Besides the carbonyl group, carbohydrates have got the OH hydroxyl group. And because those OH groups are a lot, we call it polyhydroxy. So it can either be a polyhydroxyaldehyde or polyhydroxy ketone. And what happens is that when you react with these carbohydrates with water, they will be broken down to be able to form their constituents. Now, carbohydrates, specifically more the monosaccharides, they carry the formula CH2O with N. So any structure that is going to obey this formula, you are going to refer to it as a carbohydrate. We classify carbohydrates, number one, depending on their size. For example, you'll be looking at how many carbon atoms those carbohydrates have. You have got those that have got one carbon atom, two carbon atoms, three. So of importance to understand is from three. We call those trioses, four will be the tetroses, five, the pentoses, six, the hexoses. And then you can also classify these monosaccharides, not looking at the number of carbon atoms, but on their actual size. So I've got simple sugars, which we call the monosaccharides, and then we have got the disaccharides, the oligosaccharides, and the polysaccharides. So what are the general functions of carbohydrates in the body? Number one is that they form the dietary staple. So for example, sugar and starch. When you get to when these carbohydrates undergo oxidation, they are going to produce energy that we need in the body. Besides that, some of these carbohydrates don't dissolve, they are insoluble. And so you are going to see them being used in the structure of the cell for protection. You also see them like in bacteria and in plant cell walls, and also in connective tissue. We'll discuss them. Other carbohydrates will actually be found on the joints acting as lubricants. We'll hear of what we call hyaluronic acid. Specific carbohydrates that contains some other molecules, they are going to act in cell to cell recognition. You understand cell to cell recognition a lot as we get to discuss in physiology and in anatomy cells. Now, some of these carbohydrates are actually going to be involved in adhesion. Adhesion is one cell being able to connect to another cell. And also in blood clotting, immune response, and wound healing, you are going to see carbohydrates there. So now let's discuss each one of these carbohydrates. We'll start with the monosaccharides, and then we talk about the disaccharides, and then the polysaccharides. Now, monosaccharides are simple sugars. They only have a single polyhydroxyaldehyde or ketone. So I've got only one aldehyde with a lot of OH groups, or one ketone with a lot of OH groups. And the most abundant of all the monosaccharide is D-glucose, which is also called dextrose. Now, monosaccharides can be classified depending on the number of atoms and the position of the carbonyl group. On the number of atoms, you are going to have three carbons, trioses, four carbons, tetroses, five pentoses, just like that. And then on the position of the of the carbonyl group. If it is at the end of the chain, that's an aldehyde or an aldose sugar. On the middle of the chain, we call that as a ketose sugar. For example, you can see this is an example of a triose because this carbohydrate contains the three carbon atoms. If the carbonyl group is at the end of the chain, we call that as an aldose sugar. Specifically for three carbons, we call that as glyceraldehyde. And then if the carbonyl group is on the middle of the chain, this is a ketose sugar. Specifically, this is known as dihydroxyacetone. Okay. So when you have got monosaccharides with more than four carbon atoms, five, six, seven, these will end up forming a ring. So that's important. Why do they end up forming rings? That is just to maintain stability. Monosaccharides are usually colorless. They are also water soluble because they have got all these OH groups which can interact with water. They are crystalline solids. Crystalline, the way you see sugar crystals, right? And then some of them are actually sweet. So starting with the trioses, remember these are monosaccharides we're talking about. So here I'm mentioning the aldehydes for each one of them and the ketone. So for the trioses, the aldehyde is glyceraldehyde. The ketone is dihydroxyacetone. Why do you need to know this? Because sometimes in your test or your exam, you just see questions that will come. The following are examples of ketones except, or the following are aldose sugars except. So you need to know. So these are the trioses, the aldehyde, and the ketone. And then for those which have got four carbons, tetroses, the aldehydes, you can see we have got erythros and threose, and then for the ketone we have got erythrulos. Now when you see something that has got u, likely that's a ketone. For example, you can see erythros, erythrulos. So those two are actually the same. It's just that the other one is a ketone. And then threose also is an aldehyde. Same with five carbons, you can see aldehydes and ketones. For example, we have got ribose or ribose. The ketone is ribulose. And then we have got xylose, that's an aldehyde. The ketone is xylulose. You can see those. And then we have got arabinose, is also an aldehyde. Lixose is also an aldehyde. So mostly ketones we are going to see an AU somewhere in the name. And then for the hexose sugars, those that contain six carbons, we have got the aldehydes like allose, outros. The common ones is glucose, mannose, and galactose. But you also have things like gulose, aldose, idose, talose. But look at the ketones. You have got sucrose, fructose. This one is common, right? You did it also at first year. Sobose, Tagatos. But the most important ones you need to know here, glucose, mannose, galactose. These are aldehydes. And then for the ketone, we have got fructose. Now, when you look at all these monosaccharides that we've mentioned here, monosaccharides can end up forming isomers. Now, isomers are just different ways in which you are going to write the structure of the same compound. So you have got the same molecular formula. For example, all monosaccharides, all hexose sugars have got the formula C6H12O6. But then you can write different structures, and that forms what we call isomers. So isomers are compounds that have got the same chemical formula but different structures. What are examples of isomers? You have got fructose, glucose, mannose, galactose. These guys all contain the formula C6H12O6. They are all exo sugars. But then their difference is going to be, out of all these, fructose is an aldehyde. Glucose, mannose, and galactose, these uh, are aldehydes. Fructose is a ketone, sorry. Right? So those are isomers. Same formula, different structures. We don't care how the structures are arranged. But we also have what we call epimers. Epimers are carbohydrate isomers that differ in configuration around only one specific carbon atom. Okay, only one specific carbon atom. For example, you can see mannose, and glucose, they have got the same formula. But the only difference is at carbon number 2. You can see for glucose, the OH is on the right. For mannose, the OH is on the left. So it's important for you to understand where the difference is for glucose and mannose. We call them epimers because they only differ at carbon number 2. And then for glucose and galactose, all the carbon atoms and the way they are bonded to atoms is the same except at carbon number 4. For glucose, the OH is on the right. For galactose, the OH is on the left. So glucose and galactose, they are epimers at carbon 4, while glucose and mannose are epimers at carbon number 2. So that's what we need to know. We also have what we call enantiomers. You understand this even more in organic chemistry. Now, enantiomers are mirror images of each other. For example, when you get D-glucose and L-glucose, these are exact mirror images of each other. If at carbon number 1, the OH is on, if at carbon number 2, the OH is on the right for the D, for the L, the OH is going to be on the left, just like that. So the D isomer and the L isomer, we call them enantiomers. And the most common form of glucose is the D isomer. Now, these carbohydrates, they can actually also end up forming cyclic compounds, and that is where you see structures appearing like this because hexo sugars can end up forming cyclic compounds. A cyclic compound that looks like an hexagon, six-membered, we call it a pyranose ring. And for the hexo sugars, all hexo sugars form a pyranose except fructose. Fructose, when it forms a ring looks like a pentagon, and we call that as a furanose, not pyranose, but furanose. That's what we need to know there. So, when you are naming glucose, so when glucose forms a ring, it's a pyranose ring. But again, you also determine what we call stereoisomerism. Stereoisomerism is talking about the arrangement of an atom at a given point, okay, at a given carbon atom. How are the arrangement of atoms there? So for glucose, you look at carbon number 1. If the OH is down at carbon number 1, we call that as alpha glucose. But because glucose forms a pyranose ring, we can also call that as alpha glucopyranose. All ring structures that you see, it is only the D form that can form the ring structure. The L form can't form the ring structure. So that's why we're calling it as alpha D glucopyranose. We can just say alpha D glucose. For the fructose, it is not a pyranose, it is a furanose. So it is alpha D fructofuranose. This was supposed to be fructose. How did we know it's alpha? Because OH at carbon number 2 is down. Same with also galactose, you also follow the same way. Now, having understood all these monosaccharides, before I proceed, is there any question on any one of these monosaccharides? Any question? So, after understanding these monosaccharides, what happens is that these monosaccharides can form derivatives. What are derivatives? Derivatives are compounds that are formed when you get a monosaccharide. You substitute like one of the atoms that the actual monosaccharide has with another group of atoms or with another atom. For example, you can substitute the OH groups with another group, or you can get the molecule, that monosaccharide, and oxidize it, and it is going to end up forming a carboxylic or a carboxyl group. Okay, so when you get a carbon, you can see that a carbon, you CO, you have got a carbon with only oxygen. If oxidation happens, you are going from COO. What is oxidation? Oxidation is a gain of oxygen. So you can see that this guy has gained oxygen. That's why we are saying this process is oxidation. So the first one that we want to understand is when monosaccharides undergo oxidation, they can form, for example, aldonic acid. So what is an aldonic acid? An aldonic acid is the product that happens by the oxidation of the aldehyde group at carbon number 1, or of these hexo sugars or monosaccharides. Please take note, it is oxidation number 1. For it to form an aldonic acid, what should happen is the oxidation. Besides that, the oxidation that is happening is for the aldehyde, specifically at carbon number 1. That's how the question can come on this one. So for example, when you get glucose, you oxidize it at carbon number 1, it forms that carboxyl group. It becomes a gluconic acid. Remember, this is aldon. All these derivatives will be aldonic acids. If this was galactose, it would become galactonic acid. Fructose, but fructose is not an aldehyde, so it won't form these aldonic acids. Hope you get the sense. Glucose to form gluconic acid. They can give an this one. They say in the exam they just give gluconic acid. How do you get gluconic acid? By oxidation of carbon number 1 of glucose. Now, what is the use of aldonic acids? These sugar derivatives are used in many ways in the food industry as gelling and the solubilizing agents. They are going to be used to make other molecules soluble or other molecules to dissolve. They'll also be used in cosmetics as antioxidants, preventing oxidation. Now, oxidation is a very big process that leads to the injury in our cells. So that's why we need antioxidants, because once you oxidize compounds, you are going to end up producing what we call reactive oxygen species, and this can destroy our cells. So these aldonic acids will be used in cosmetics as antioxidants, in the food industry as gelling and solubilizing agents. The second derivatives we need to know is known as uronic acid. Differentiate them. This is was aldonic acid. This is uronic acid. Uronic acid happens because of the oxidation of the hydroxyl group, not the not the aldehyde. We are not oxidizing the carbon number 1, the functional group. We're oxidizing the hydroxyl group, which is at carbon number 6. And what are we oxidizing it to? To carboxylic group. It's the same like aldonic acid. For example, when you get glucose, you oxidize it at carbon number 6. You are going to form glucuronic acid. Differentiate this one, which was gluconic. That was an aldonic acid. This is a uronic acid, glucuronic. Okay, so we are just getting the uronic, and then we put Gl. It becomes glucuronic. And this was aldonic. What we just did is we removed the ALD, and then we put GLU, glucuronic acid. So what do we use uronic acid for? Uronic acids, for example, we have got what we call glycosides. Glycosides, these come from D-glucuronic acid. So the same glucuronic acid, which comes from the oxidation of glucose at carbon number 6, can also form what we call glycosides. And so these ones, the glycosides, besides coming from D-glucuronic acid, they also come from steroids. These will actually appear in the urine of animals as part of the metabolic product. So uronic acids, they are going to be used. What happens is that because the, these are acting as excretory product, foreign toxic substances are going to be converted in the liver to these glucuronides before they can be excreted in urine by conjugation. Okay, so glucuronic acid is a major component of connective tissue also. So we are saying toxic substances will be converted to this type of acids before they can be excreted in a non-harmful way. It's also a component of connective tissue. You understand connective tissue as we discuss anatomy. So we've talked about aldonic acid, uronic acid. We also have what we call aldaric acid. Now, aldaric acid is a compound that is formed when there's oxidation at both carbon number 1 and carbon number 6. Carbon number 1, we are oxidizing the aldehyde. Carbon number 6, we are oxidizing the hydroxyl group because the carbon number 6 we have got OH. Okay, so we're oxidizing both of them to carbonyl groups, carbon number 1 and carbon number 6. We are oxidizing both of them to carboxyl groups, sorry. For example, you can have glucose, you oxidize it to what we call glucaric acid. Can you see how it is ending? That's ending in caric because it's an aldaric acid. And this glucaric acid, it acts as a chelating agent. A chelate is a compound that binds metals. For example, imagine someone has undergone what we call uh, heavy metal poisoning. You want to remove that metal, so you need something that will be able to bind to such a metal. So a compound that binds to metals, we call it a chelating agent. So it binds calcium and magnesium ions. So that's aldaric acid. We also have what we call alditors. Now, alditors are not formed by oxidation. We've talked about aldaric acids, aldonic acids, uronic acids. We've said that those happen by oxidation. Alditors happen by the reduction. When you get an aldose sugar, you reduce it to form now a hydroxyl group. For example, can you see at carbon number 1 there you have got the aldehyde functional group, that double bond with oxygen. So you reduce it, it becomes an OH. That's known as an aldito. So specifically when you reduce glucose, it will form an aldito which we call sorbitol. Okay, so sorbitol is an aldito. It's formed by reduction of glucose. We need to understand that. So what do we use auditors for? They are used in the food industry as thickeners and sweeteners. They are also used as sugar substitutes for diabetes patients. So someone that has got diabetes, who have got a lot of sugar in the body, you can get to substitute that sugar that they have, that is leading to a condition of high sugar in the body. We call it hyperglycemia. So you can substitute the sugar with these auditors. We also have what we call deoxy sugars. For the deoxy sugars, these are the monosaccharides in which the hydroxyl group is reduced to a hydrogen. The common one is the ribose. At carbon number 2, when you get this ribose, you remove an oxygen, you just remain an hydrogen. We call it deoxyribose. That's a deoxy sugar because it has lost an oxygen. And the deoxy sugars are used in the formation of purines and pyrimidines, which are used in the formation of DNA. We also have what we call amino sugars. Now, sugars, what happens is that at carbon number 2 of these carbohydrates, we have got the OH. For example, you can see glucose here. Carbon number 2, we have got OH. So what we do is that at carbon number 2, this OH, we are going to replace it with an amino group, that is NH2. Okay, so the name changes from glucose to N-acetyl glucosamine. And then what happens is that this N-acetyl glucosamine will quickly be will quickly add another group to it. Okay, we'll quickly add another group to it. You can see the carbon double bonded to oxygen and then see H3. And so the name changes everything. So the first thing is glucose. So the first thing, please let me clarify this. When you get glucose, you add an amino group, it becomes glucosamine. Okay, glucose, just add amino, so glucosamine. Now, when you get this glucosamine, you add an acetate. This is the C double bonded oxygen and then 33. It becomes N-acetyl glucosamine. Why are we calling it N? Because we have gotten the acetate and added it to the nitrogen, so that is N-acetyl glucosamine. Now, the N-acetyl glucosamine is very important. Where is it found? It is found as part of the cell of the bacteria, if you see that in equation. And then we also have what we call sialic acids. Sialic acids contain 9 carbons and they are usually present as terminal residue of glycan and glycoconjugates. You Like they are found at the end of glycans, glucose molecules, and the conjugates of glucose molecules. You understand, of course, specifically glycans, these are just glucose molecules that have been attached together to form a long chain. So they'll be found at the end of that. And an important one, an important sialic acid is any acetyluramic acid. And this guy, any acetyluramic acid, is formed when any acetyl manosamine reacts with the phosphoenolpyruvate. What are the function of sialic acids? They play an important role in the communication between cells, also stabilizing the cell membrane. They also help in binding and transporting ions and drugs. Okay. Also stabilizing the formation of proteins, like for example, when you are forming an enzyme to be able to stabilize the structure, you need also sialic acids. They protect molecules and cells from attack by proteases or glycosidases. Proteases, these are enzymes that break down proteins, and glycosidases are enzymes that will break down uh, carbohydrates. This is the structure of these sialic acids, not something you should master. So those are monosaccharides and the sugar derivatives that comes from the monosaccharides. Now the disaccharides, something simple. Disaccharides are formed by two monosaccharides. Okay, for example, you have got maltose formed from combination of two glucose molecules. Lactose, also called milk sugar. This one is formed by a combination of glucose and galactose. Sucrose is formed from glucose and fructose. So we get any two monosaccharides. For example, here glucose plus glucose, you can see carbon number one of one glucose, we call that as a hemiacetal with the OH. Now the carbon number four, we are only getting an hydrogen there, and then that. So it is the OH from carbon number one with the hydrogen from carbon number four. So water is released in this process. So when you're forming disaccharides, polysaccharides, oligosaccharides, there is a condensation reaction because you lose water. So to break down the the disaccharide, you need to react it with water. That's when you form the constituent monosaccharides, and you call that hydrolysis. So sucrose, which is the table sugar, plus water, we call that as invert sugar. And some of these disaccharides are actually going to be reducing sugars, others are not reducing sugars. So for the reducing sugars, what happens is that, for example, can you see in this one, carbon number one reacted with carbon number four. So carbon number one of the other glucose molecule is still free. So because the carbon number one, which contains the functional group, is free, it can still reduce other substances. That's why we call them reducing sugars. The only disaccharide for now that you need to know that is not a reducing sugar is sucrose. Why is it not a reducing sugar? Because sucrose is formed from glucose and fructose. Glucose, the functional group, has carbon number one. Fructose, functional group, is at carbon number two. And when you are forming sucrose, it is carbon number one of glucose and carbon number two of fructose which react. The meaning of that is that both functional groups have reacted. So you don't have a free functional group that can reduce other molecules. That's why sucrose ends up being a non-reducing sugar. Okay. So let's talk about lactose a bit. Lactose, formed from glucose and galactose, can be broken down by an enzyme which we call lactase. But in some people, there is deficiency of this enzyme lactase. So what happens that when you drink milk, that milk can't be broken down because monosaccharides, I mean carbohydrates, are only absorbed as monosaccharides. So the meaning is that if you take sugar, if you take milk, and then you can't break down that disaccharide, which is the lactose, to glucose and galactose, it means that you can't absorb that milk. So what happens in that case is that the milk will just be in your intestines. Because milk has got, though it's a carbohydrate, it has got these OH groups. They'll be attracting water. They'll be attracting water. So water will be moving from the cells going into the intestines, and that leads to diarrhea and flatulence. Okay, that that feeling of removing, like your stomach or your intestines is moving, and then it's making noise, and then you are also having, I don't want to use the actual term, okay, removing gas like that. So lactose is because it has accumulated and then water is going to it. So this one we call it osmotic diarrhea because water is being poured from the cell to the intestines. So that is what happens in this condition. Okay. So there's going to actually be malabsorption of this lactose. You can't absorb it. Not really that we can't digest it. Okay, we can say so. Yes, we can't break it down in that case, right? And so it can be absorbed and then it attracts water. The polysaccharides, these ones we also call them glycans. Now we also have oligosaccharides. Oligosaccharides are made up of carbon, like from 3 carbons to 10 carbons. We call those as oligosaccharides. Beyond that, we call them polysaccharides, also known as glycans. Now, for polysaccharides, we have got polysaccharides that are made up of only a single type of monomer. For example, only glucose alone. We call those as homopolysaccharides. And then you have got those that are made up of different types of monosaccharides. For example, glucose and another thing. So we call those as heteropolysaccharides. Now, polysaccharides have got different functions. For example, the homopolysaccharides, you have got those that are going to be used to store uh, as a storage form of energy. So, examples is starch, which stores the energy in plants, glycogen, which stores energy in animals. And then you have got some which are used in the structures. For example, cellulose and chitin. Cellulose is found in the animal cell wall. Chitin is found in what we call the exoskeleton. Sorry, cellulose, not animal cell or plant cell. Animals don't have a cell wall. So chitin is found in the exoskeleton. For example, a grasshopper has got an outer skeleton. It's made up of chitin. Now the heteropolysaccharides, these ones will form what we call extracellular support for organisms. You understand the extracellular thing when we discuss anatomy again. What happens is that our bodies are made up of cells and what we call extracellular matrix. Think of extracellular matrix as what holds the cells together. Just like you have got bricks, and then you have got mud, right, to hold those bricks together. So the bricks will be the cells, and then the mud, that will be the extracellular matrix. So polysaccharides will provide that extracellular support for organisms. So you can see the homopolysaccharides, they are made up of one type of monosaccharides, the hetero two different. And for this, you have got some which are straight and some which are branched. So starting with the homopolysaccharides, they are the most important storage polysaccharide, and these are starch in plants and glycogen in animals. And these guys occur inside the cell as clusters or granules, like they are found together. The starch molecules will be found together like that. Now these guys, where you see them, because they contain those OH groups, right, the starch and glycogen, they attract a lot of water. So they are heavily hydrated because they have got a lot of hydroxyl groups. And because of this, that is why they are actually not a very better form of storage, because once you store energy in terms of starch and glycogen, there's also a lot of water there. Now, starch is found in things like potatoes and in seeds. Starch is made up of two types of glucose polymers. You have got amylose and amylopectin. Amylose is not branched. It only contains alpha 1, 4 glycosidic bonds. Amylopectin is branched. It contains alpha 1, 4 and alpha 1, 6 glycosidic bonds. So take note, the branches in starch, specifically amylopectin, happen after 24 to 30 residues. Okay, 24 to 30 residues. And the amylopectin and amylose can be broken down by the enzyme which you call alpha amylase. So you can see amylose is straight, amylopectin is branched. The branches are occurring after 24 to 30 residues. The residues are the monosaccharides. Glycogen is similar to amylopectin. It is also branched, but the branches are more frequent, like they happen for every 8 to 12 residues. You can see it's also more compact than starch, like the molecule is more packed in comparison to starch. Glycogen is most abundant in the liver and skeletal muscles because that's where most of the carbohydrate is going to be stored. In the liver cells, which we call hepatocytes, glycogen is going to be found as granules. And these granules, like the glycogen is found packed together, is going to be in such a way that it contains the enzymes that are responsible for breaking down glycogen and also for creating glycogen. Can see that's the structure. So those are the important homopolysaccharides. And then for the heteropolysaccharides, these are made up of different monomers. The first one you need to understand is peptidoglycan. Now, peptidoglycan is found in the cell of bacteria. Please don't forget that. And it contains alternating units of beta 1,4 linked structures of N-acetylglucosamine and N-acetylmuramic acid. Please don't forget what makes up peptidoglycan: N-acetylglucosamine and N-acetylmuramic acid. Okay, and these have got beta 1,4 bonds. And what happens that if this is, let's say the one in green is the N-acetylglucosamine, actually that is N-acetylmuramic acid, and then the one in red is the N-acetylglucosamine. These guys are going to be connected. So if you have got one chain like that, and then you have got another chain, you are going to connect these different chains using short peptides. Peptides, these are amino acids. Those are the ones that will be able to connect these together. And so what happens is that in our, because these peptidoglycans are found in bacteria, right? And our Ts actually have got an enzyme which we call lysozyme. The lysozyme, what happens is that when you shed your tears, let's say for example, something entered your ear and maybe there's a bacteria there, the lysozyme is going to be able to break down those glycosidic bonds between N-acetylglucosamine and N-acetylmuramic acid. And so when you break down those bonds, what happens is that you are destroying the structure for the bacteria cell wall, and that is how you can be able to be protected from bacterial infections. Okay. Now, some drugs like penicillin and other drugs that fight bacterial infections, which we call antibiotics, they are going to kill bacteria by simply preventing the synthesis of these cross linkages by these polypeptides, these short peptides that we can see. So penicillin and other antibiotics will stop the formation of those bonds. And so what will happen is that water can easily enter into the bacteria and then it will be able to burst. So that's how we'll be able to treat the conditions that you give a medicine, penicillin. Penicillin is a painkiller. Now, so those are peptidoglycans. Please don't forget what makes them up, and then don't forget about the close linkages and what happens with the lysozyme. Glycosaminoglycans, we call them GAGs or GAGs. Glycosaminoglycans. Please take note of the spelling. Sometimes can come and then you put GU, GL, especially if it's an online test. But more likely you will not have any online test. So glycosaminoglycans are composed of repeating units of N-acetylglucosamine or N-acetylgalactosamine with mostly uronic acid. So take note of that. N-acetylglucosamine or N-acetylgalactosamine with uronic acid. So GAGs or glycosaminoglycans, these are used in the extracellular matrix. They are the ones that will be holding together the cells. Okay. and they are going to also be forming a mesh like that to be able to hold the cell together, and also structures like collagen, elastin, and fibronectin. Now, glycosaminoglycans are of different types depending with what type of uronic acid you have used, or the combination of the one I've put one there with a two. For example, we have got what we call hyaluronic acid. It's a glucosaminoglycan. It's made up of D-glucuronic acid. Remember, we said an example of uronic acid is when you get glucose, you oxidize it at carbon 6, you are going from glucuronic acid. So glucuronic acid is actually a uronic acid. So if you get glucuronic acid and then you get any acetyl glucosamine, you would have formed the hyaluronic acid. You find in a question they'll just give you what makes up hyaluronic acid in the exam or in a test. So those are the components. And then so hyaluronic acid, we also call those as hyaluronates. N-acetyl glucosamine and D-glucuronic acid. So where do you find hyaluronic acid? These are going to serve as lubricants in the synovial fluid, like on our joints. Okay, and also the vitreous humor of the vertebrae eye. So vitreous humor, this is the fluid that you find in the eye. Now of the vertebrate eye. So vertebrates, these are animals that have got a backbone, right? So the fluid that you find in the eye is actually made up of this hyaluronic acid. So it's a jelly-like fluid. So it's very important for extracellular matrix of cartilage and tendons. You also have what we call chondroitin sulfate. Now, chondroitin sulfate is made up of the same D-glucuronic acid, but instead of using N-acetylglucosamine, you use N-acetylgalactosamine. That's chondroitin sulfate. It's also going to contribute to the tensile strength of the cartilage, tendon, ligaments, and the wall of the aorta. Tensile strength is when you get to pull this thing, it can still come back to itself. You also have what we call dermatan sulfate. This one is formed by N-acetylgalactosamine, but the uronic acid we are using is iduronic acid. This one contributes to the permeability of the skin and prevents and also is in the blood vessels and heart valves. Permeability is the capacity for you to be able to pull the skin and it can still come back, right? Keratin sulfate is made up of N-acetyl glucosamine and galactose. Now, keratin sulfate is present like in the cornea, cartilage, bone. Keratin makes up dead cells. For example, our skin is dead, like the outermost layer of the skin is dead. Okay, and also nails, hair, clothes. So those are the components. We also have what we call heparin. Heparin is made up of N-acetylglucosamine with either glucuronic acid or iduronic acid. Now, heparin is an anticoagulant. It prevents the clotting of blood. It's actually released by our white blood cells. Okay, so it prevents blood clotting. Imagine your blood starts to clot while you are walking. What will happen is you will die. So heparin is doing that work. We also have what we call glycoconjugates. In conclusion, glycoconjugates, these are formed uh, by just getting a glucose molecule and then getting to add something to it. Okay, for example, we have got what we call proteoglycans, glycoproteins, and the glycolipids. Look at this. Proteoglycan means protein and the carbohydrate. Glycoprotein means the carbohydrate and protein. So what's the difference? So you need to know the difference between proteoglycan and glycoprotein. And then we have got glycolipid, carbohydrate with a lipid. Proteoglycan is made up of glycosaminoglycans and a protein. And you can see, for example, the glycosaminoglycan that we are using here is the hyaluronic acid with a protein. Okay, and these are joined by covalent bond. The glycosaminoglycan commonly forms the greater part of the proteoglycan. Like that's the core. This is the one that is on the middle. and it's the glycosaminoglycan that is the main site for the biological activity. So these proteoglycans are a component of connective tissue, like in cartilage, you see them there. And then glycoprotein is made up of the carbohydrate part of glycoprotein is the oligosaccharide, and then you have got a protein. For proteoglycan, the carbohydrate part was specifically glycosaminoglycan. Here it is just an oligosaccharide. Okay, so glycoproteins are found in specific organelles such as the Golgi complex, secretory granules, and lysosomes. These guys, glycoproteins, many of the proteins secreted by the eukaryotic cells, are actually glycoproteins. For example, antibodies and hormones like luteinizing hormone, follicle stimulating hormone, those are glycoproteins. And then you also have glycolipids. This is just where I've got a lipid and a carbohydrate. Okay, for example, this structure here. An example of a glycolipid is what we call ganglioside. What makes up ganglioside is the sialic acid, that's the carbohydrate part, and the glycosphingolipid, that's the lipid part. So you can just see, I've seen this one in one of the test papers. Ganglioside is made up of that sialic acid and glycosphingolipid. So these guys act as in cell to cell recognition and communication, and also specific receptors. Receptors are the ones that will be able to pick up information from the external world so that the body can interpret. We also have what we call lipopolysaccharides. Lipopolysaccharides, these are going to be on the surface of the cell. Lipopolysaccharide, these are important targets for antibodies produced in response to bacterial infection. So, if let's say I've got a bacteria in the body will produce something to fight against that bacteria. We call it the antibody. So, the antibody will actually come and bind to the lipopolysaccharide, and these guys are actually found in gram-negative bacteria, not gram-positives. Okay, so that is it on the first part to this carbohydrates. I'm going to send some practice questions for you to try out. We'll see if you can be able to answer from what we've done today.